So let's start. So very good evening to everyone. How are you children? All are doing well? Great. Am I audible? Hello, 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 hello. Sound is coming? Yes. So can you tell up to what we discussed in the last class? Can you pin up to where we discussed? Hello. So cell the unit life over? Giant chromosome I told or not told? Giant chromosome told or not told? Simply write yes or no. Giant chromosome, not answer coming. Cilia and flagella completed. After that, nucleus not completed. Nucleus also completed, na? I hope in your batch. No? Are you sure? How can nucleus not completed? Nucleus I have completed probably. Centriole, nucleus, these I drawn probably was missing in class. You missed the class, probably last class. Nucleus over, right? Yeah, I am very sure it is over. Yeah. Chromatin, chromosome over means nucleus over, na? Nucleus I drawn, diagram, everything, right? I just recall. Okay, let's start. So I'm starting with giant chromosome. I just want to give you some information about giant chromosome. It is not in NCERT, but if you go for last 10-year paper, 4 to 5 questions came from the giant chromosome, right? That is the reason I am teaching it. Okay? So if you talk, but not much detail I will tell. If you talk about giant chromosome, it is of two types. One is polygyny, or it is also called salivary gland chromosome. Salivary gland chromosome. And another one is Lambrus chromosome. Second type is what? Lambrus chromosome. Lambrus chromosome. Isn't it? So there are two types of giant chromosome we will discuss. So salivary gland chromosome or polygyny discovered by Balbiani. And if you talk about Lamprus chromosome, it is discovered by Fleming. I am going to say only that information from where there is a possibility to ask the question. Not much. Right? Second thing I am writing, these polygyny or salivary gland chromosome found in salivary gland of Chironomus larva. Salivary gland of Chironomus larva, or you can say, or better to write, and Drosophila. Where these Lambrus chromosome present? It is present in oocyte of, found in oocyte of vertebrate. Vertebrate, exception is there, except mammal. And this is present in which one? In diplotin stage. In diplotin stage of prophase 1. So which one is present? Many times this line asked in the competitive exam, NEET exam or All India PMT exam. Okay, so diplotin stage. It is found in which one? Salivary gland. So polytiny found in salivary gland, and Lambrus chromosome found in which one? Oocyte. Got the idea? Keep in mind. It is very important information. Next, if we talk next, so here you will find it produced due to, I am writing about Lambrus, so polytiny, produced due to, due to no segregation of chromosome, no segregation of chromosome after DNA replication. After DNA replication. Just try to understand. Normal condition is what? I'm just telling about, once again, try to understand polytiny. Normal condition, what's happened in normal chromosome? This is in G1 phase. After S phase, it become like this. And in NA phase, what happened? Both get segregated and move toward opposite pole. It is normal condition. What happened here abnormal? After S phase, sister chromatid formed, but they not segregated. So what's happened? One chromosome getting many, which one? Chromatid joined with it, and which make it giant. So many hundred of chromatid attached with the common centromere. So number of chromosome is more, chromatid is many. Are you getting the idea? Chromatid is many. Chromosome is one because number of chromosome depend on number of centromere. Centromere is one, but one chromosome it been hundred of chromatid. Now just try to understand. 100 chromatid, same type of 100 copy of DNA is there or not? And every DNA have gene. So the gene have a hundreds of copy. Same gene have a hundreds of copy. Gene produce the protein. So protein produce hundreds times or not? An example. Are you getting the idea? So what are the advantage of this? You got the idea? Understanding the things clear in the mind? And now from here, one thing more going to ask. Here, one thing more going to ask. What you will find here in the chromosome, there is a ring-like structure. I am just drawing a diagram. Suppose that this is a Lambrus chromosome, and here ring-like structure are present. So this is which one? Bal-Biani ring. Examiner ask a question. Bal-Biani ring present in Bal-Biani ring, and these Bal-Biani ring present in Lambrus chromosome. And these Bal-Biani ring is made up of what? Chemical composition of Bal-Biani ring. These Bal-Biani ring is what? Chemically DNA. Chemically DNA. And DNA participate in which one? Protein synthesis. Protein synthesis. So what are the probable question going to ask from here in the exam is, first question examiner ask, let me tell you in the question format. Let me tell you in the question format what the possible question going to ask. First question they will ask is, polytiny or salivary gland chromosome present in salivary gland of Chironomus larva or Drosophila. Right? Getting the things? Now, how they are producing? Method of production is what? After DNA replication, segregation not occur. So with many sister chromatid attached with the same centromere, so it become a giant. When you look the structure here, ring-like a structure present in the chromosome, and this is called Balbiani ring. That is chemically DNA and have a role in protein synthesis. Got the idea? So that the information minimum you keep in mind, right? So these things help in, these more amount of production help in which one? Conversion of larva into pupa. So larva convert into pupa with the help of the protein produced by these Lambrus chromosome, these polytiny chromosome. Got the idea? Now we are going to talk about which one? Lambrus chromosome. I already told you. Discovered by Fleming, present in oocyte of vertebrate except mammal, and are present in reported in which stage? Diploid in stage of the prophase 1. If you look the Lambrus chromosome, it look like brush of lamp. In village area where electricity is not there, people use the lamp, and this is the lamp cleaning brush. So it appears like a lamp cleaning brush. So it is a which one? Lambrus chromosome. What is most important? Present in oocyte. Here, what is most important? Present in salivary gland in case of polytiny. Done. Over. So this is about the giant chromosome. Over. Cell, the unit of life chapter over. Now we are starting a new chapter, and that is called biomolecule. Right? Have a look. Biomolecule. Have a look here. If we talk about this chapter, biomolecule, you know there is a diverse type of organism present in your surrounding. Right? Animal, plant, bacteria, fungi, right? So they have a different appearance. Beside of that, they are made up of cell. That I told in the previous chapter. But if you talk about chemical composition, elemental analysis, you will find they are also made up of same kind of elements. If I am telling you, listen me carefully, कि what are common between, if I ask a question here, कि what are common between living and non-living? What are common between living and non-living? What the answer will you give? In living and non-living, what is common? One thing you can say, na, we both are made up of matter. This styler, pen, is a matter. I am standing as a human being, also a matter. Got the idea? So both living and non-living are made up of matter. Right? Now, if you go for the matter, you better know matters are made up of atoms and molecules. Got the idea? If you go for the analysis of a non-living material and living being, you will got a very surprising result. You will find what are the elemental composition present in our body or a plant body. Same elemental composition present in the earth crust. Means our body is made up of carbon, hydrogen, nitrogen, oxygen, phosphorus, sodium, calcium. Getting the things or not? In earth crust, in soil, in bricks, also same things are there. Carbon, hydrogen, oxygen, nitrogen. So we are made up of almost same kind of elements. I'm using the almost, means usually most of the cases. Not exactly 100% same, almost. Term we use for similar. So we are made up of what? Similar kind of chemicals. Almost same kind of chemical. You just try to understand, but how we differ from the non-living. So percentage of a particular element in a particular mass is different in our body as a difference in earth crust. So you just look about the elemental analysis. You just look about elemental analysis. So I am writing here elements. Here I am writing percentage in which one? Earth crust. Then I am writing what? Percentage in human body. Human body. And I am writing about the name of element. So I'm writing the element like oxygen, carbon, hydrogen, nitrogen. Got the idea or not? Then I'm writing about which ones? Silicon, then I'm writing calcium, then I'm writing sodium. So that much I'm writing. So do you know how much percent oxygen present in the human body? It data of your NCERT. In human body, oxygen is how much percent? 65%. Right? Carbon is how much? It is 18.5%. If you talk about hydrogen, NCERT wrote 0.5%. Here is a data glitch. It is wrongly written in NCERT. If you talk about nitrogen, nitrogen present is how much? 3.3%. Got the idea? And these oxygen present in earth crust is how much? 46.6%. Carbon present is how much? 0.03%. Hydrogen present in how much? 0.14%. And nitrogen is very little. Very little. Getting the idea? Is it clear in the mind? And if you talk about silicon, it is present 27.7%. Here it is negligible. If you talk about calcium, it is present somehow 3.2%, approximate. Check the data of the NCERT. Here it is present how much percent? 1.5%. Here sodium it is 2.2% approx. Here it is present 0.2%. Got the idea? Just match with the NCERT. It may be some fluctuation. So check it. Got the idea? Got the idea? So these are the comparison I did in the elemental compositions of which one? Earth crust and human body. What you are finding? These are present more in which one? Human body. And these are called major 4. I am also telling you. These are called major 4. In our body or any living beings, oxygen, carbon, hydrogen, nitrogen present in the maximum amount. And any other book other than NCERT wrote hydrogen is more than nitrogen. And if you go for any other books other than NCERT and this level textbook who are based on NCERT, they are written what? Hydrogen is 8 to 10%. Got the idea? Because these major 4, anywhere you can Google it also, it is 95% approx. So oxygen, carbon, hydrogen, oxygen, if we are keeping hydrogen 0.5%, it is not coming 95%. It is coming something 86%. Getting the idea or not? Right? So what is the glitch? Data glitch in the case of hydrogen in the NCERT. Got the idea? Now anyhow, now I want to say you one thing, that is what? Silicon, calcium and sodium is present more in earth crust in compared to which one? Human body. Got the idea? So what you find with this data? Some of the element present more in human bodies and living organism in compared to earth crust, and some of the element are present more in earth crust in compared to, and there is a bigger differences shown by which one? Silicon. Silicon present in the earth crust is 27.7%, and in the case of our body, it is negligible amount of silicon are present. Are you getting the idea? Understanding the idea? Is it clear in the mind or not? And even NCERT going to prove it is a data glitch here. How? When you read the NCERT, NCERT wrote a line. Na? What line is written in NCERT? NCERT wrote कि in living wing, living cell, living organism, carbon and hydrogen is more abundant than non-living. So you look the data, leaving it justified here. Ki bhaiya carbon here is how much percent? Carbon here is how much percent? You just have a look. 18.5 here, 0.03. Hydrogen justification not came na. Hydrogen justification not came na. 0.14 and here you are saying 0.51. What is the difference? Even not a difference of 0.5%. So here it is justified with this line, here is a data glitch. Some typing error happened, nobody care about it. From many years it is going like that, teacher are teaching like that. Got the idea? You can go through that. You will find here is a data glitch. In some standard textbook you will find these are variable. Now I am going to move ahead. Now if anybody, now NCERT, when you read, NCERT author ask, Kiji, okay, elemental analysis you did. Now how to separate the different organic compounds present in the living beings? How to separate different organic compound present in the living beings. So when you read your textbook, what the things are there? They are writing na, ki for that take a fresh tissue like meat, liver you can say take on of organism. Another things take a fresh fruit or vegetable. So I am just taking here a what? Container. In this container, I am taking a tomato. Right. Okay, ji. This is what? Tomato. Now what I am doing? I am adding here trichloroacetic acid. I am adding here trichloroacetic acid. And then grind it. We are taking mortar, pestle and grind it. After grinding, what happened? We make a pestle. It get crushed. After crushing, you will find a soup-like a structure will form. Got the idea or not? Which kind of a structure going to form? Soup-like a structure going to form. So this soup look like tomato soup, but it is not a tomato soup because we have not taken here water. We have taken which one? Trichloroacetic acid. So when you read the textbook, they wrote here who is forming thick slurry. Now take another container. Now take another container. And here you just to filter it, because it is soup like na, just take cotton cloth or cheesecloth. Cotton cloth or cheesecloth. And pour it here. So what you will find when you are pouring it? You will find something who are smaller in size can it passing through the filter, and who are larger not passing through the filter. So who are passing through the filter is called filtrate. It is called what basically? Filtrate. So who are passing through filter is called filtrate, and who are not passing is called retentate. So filtrate is forming acid soluble pool. Acid soluble pool. And in these acid soluble pool, thousands of organic and inorganic compound present. Thousands of organic and inorganic compounds present. In which one? In these acid soluble pool. And now if we talk about retentate, it is not filtered. And this not filtered part is called which one? Acid insoluble pool. Acid insoluble pool. Who not dissolve, not in the trichloroacetic, not pass through this cheesecloth or which one? Cotton cloth. So acid insoluble pool have 4 components. It 3 are macromolecule and 1 is micromolecule. So all macromolecule present in acid insoluble pool and 1 micromolecule present in acid insoluble pool. 1 micromolecule. What the name of all macromolecule? Macromolecule name I am writing. Here it is protein. Here it is protein. Here it is what? Polysaccharide. Another is which one? Nucleic acid. Nucleic acid. And in micromolecule heading, lipid is present. Lipid is present. Got the idea? So except all these four, protein, polysaccharide, nucleic acid, and lipid, every organic and inorganic compounds are present in which one? Acid soluble pool. Except these four, all are present in acid soluble pool. If anybody going to ask you, amino acid present in acid soluble pool, glucose present in acid soluble pool, nucleotide present in acid soluble pool, except these four, everything is present in which one? Acid soluble pool. Are you getting the idea? So these are the how we are just separating the which one? Acid soluble and acid insoluble fraction. Now how to separate different amino acid protein? Definitely it is not at this standard. You will study later when you go for higher degree in the biochemistry. Got the idea? Now we are moving ahead. Now NCERT authors say if you want to know about which inorganic material are present in the living beings, how to know it? I want to know which inorganic substances are present in a plant. So what I will take? I am just taking a plant leaf. Okay, then I am going to dry it. After drying, who will come out? Water in the form of vapor. We got which weight? Dry weight. Weight along with water is called fresh weight. Weight without water is called dry weight. Then what we are doing it? Burning this leaf. Burn it. After burning, what happened? Organic compound. Organic compound will go in oxidation, and after oxidation it converted into which one? CO2 and H2O, carbon dioxide and water vapor. And what are the inorganic material present in ash? After burning you got the ash, na? If you go for which one? Ash analysis, you got different inorganic substances. So here we are not going to do how to do this analysis. So this is the method they told. In higher classes you will study about it, how to separate different inorganic substances. There is a branch in engineering. People do it there, like metallurgy. Metallurgy you study all these things, na? Separation technique. So there is a branch of engineering. 4 years they are studying how to separate the things. Got the idea? Anyhow, now we are moving on. Another aspects of the things. When further you read the lines of your textbook, your NCERT writing, for chemistry people, study is based on properties of what? Functional group. Like in organic chemistry, you study in alcohol, acetone, ether, ester, like that. But for biology people, we are studying about what? Amino acid, protein, fat, carbohydrate, lipid. So now we are starting our story with which one? Amino acid. Give your attention. So have a look, we are going to deal about amino acid. So if you talk about amino acid, it is substituted methane. Substituted methane. How? Look, I am drawing a structure of methane. This is a methane structure. Now what happened? I am just writing the structure of amino acid. See, this hydrogen substituted by R. This hydrogen substituted by NH2, that is amine group. And this hydrogen substituted by carboxylic group, acid group. Got the idea? And here it is hydrogen. So what I told, in amino acid, hydrogen gets substituted by different functional group. And this is a general formula of amino acid. So in amino acid, at least one amine group is there, another acid group is there. And that is the reason the name is what? Amino acid. Getting the idea? That is the reason the name is amino acid. Is it clear in the mind? So have a look now. So in every amino acid, that much part is constant. So each amino acid vary from each other by which one? R. Got the idea? So out of how many amino acid participate in protein synthesis? Out of 20 amino acid which participate in protein synthesis, getting the things or not? 20 amino acid participate in each amino acid differ from each other by R group. Every amino acid have a different R group. All 20 amino acid have a different R group. Getting the idea or not? You just try to understand it with the help of example. I am just writing the example here. So have a look. I am just drawing the common part first. So C, COOH, H and NH2. This is I am writing for first. C, COOH, H, NH2. Common part I am writing for second. Now here NH2, C, COOH, H. So I wrote the common part. How the R group vary here? So if in place of R it is H is there, it is glycine. If in place of R it is CH3 is there, it is which one? Alanine. And if in place of R here CH2OH is there, it is serine. So it is practically you are experiencing now. If R group is changing, amino acid is changing, and these are the common, white color one is common in every amino acid. Is it clear in the mind or not? Right. Now have a look here. Glycine is the simplest amino acid. And it is the only amino acid who don't have chiral carbon. You know na chiral carbon? When carbon attached with all 4 different functional group. Here glycine not have a chiral carbon. Reason is what? Both hydrogen are there. Able to understand? Except glycine. You see, here is a chiral carbon, na? Here amine, here methyl, here COH, here hydrogen. Getting the things or not? So except glycine, everyone have which one? Chiral carbon in it. Is it clear in the mind? Is it all right? Now we are moving ahead for further classification of amino acid. So look here. If we talk about the amino acid, I am classifying the amino acid into 3 categories. One I am writing acidic amino acid. Another I am writing basic amino acid. Another I am writing neutral amino acid. So if you talk about acidic amino acid, what are the example? Glutamic acid. What are the example? Aspartic acid. If you talk about basic amino acid, it is histidine, it is arginine, and it is which one? Lysine. Now remaining 15 are neutral. Remaining 15 are neutral. And NCERT wrote neutral example what? Valine. So I am writing this example as well. So valine is a neutral amino acid. So these are the classification of amino acid, acidic, basic and neutral. How this classification done? You just try to understand. Suppose that I am just drawing amino acid once again, the empirical formula, COOH, general formula you can say NH2 and here H. See, this is a amine, it is a basic group. and this is carboxylic acid, it is acidic group. So one base, one acid, overall neutral. One base, one acid, overall neutral. So now it's depend upon which one? R group. If R group have one more acid, so two acid, one base, acidic. If R group have one more base, then two base, one acid, then basic. Are you getting the idea or not? If R group don't have any acid and base, then it becomes which kind of amino acid? Neutral amino acid. Is it clear in the mind or not? Now I am just moving ahead. Have a look here. Now I am going to talk about aromatic amino acid. Aromatic amino acid. Give your attention. If you talk about the aromatic amino acid, what the example NCERT write? Tyrosine. Another example, what is written in NCERT? Tryptophan. Another example, what it is written? Phenylalanine. You know na aromatic compound who have benzene ring. So these all three amino acid have which one? Benzene rings. So it is the aromatic amino acid. Now I am going to talk the most important part in the amino acid. And that is what? Deuter ions. So what is deuter ion? First try to understand. Such kind of deuter ion who have both positive and negative charge is called deuter ion. Net charge in what? Positive and negative both. So net charge is what? Zero. Net charge where exists? Positive, negative cancel na? Is it clear in the mind? So I just want to write amino acid may exist as deuter ion in deuterionic form at a specific pH. And this pH is variable for amino acid to amino acid. Amino acid to amino acid. This pH is variable for what? Amino acid to amino acid. Got the idea? Now just have a look. Just try to understand. Wait. I am just writing here amino acid. Just have a look here. Here it is C. Here it is COOH. Here it is which one? NH2. Here it is which one? R. And here it is what? H. Now in aqueous solution at a specific pH. At a specific pH, I already told this pH is not constant for all amino acid. For a particular amino acid, this pH is specific. So at a specific pH, this pH range may vary. Getting the things or not? This pH may be 5, maybe 3, maybe 6. Getting the idea, understanding the idea. So in chemistry, your sir will talk about that in detail. But I am just telling you what? At a specific pH, what happened? Converted into what? Look here. C NH3 plus COO minus H and here R. So what happened? From here to here, COOH, proton attacked on which one? NH2 and converted into which one? NH3 plus. So here both positive and negative charge is existing or not? Hydrogen have electron and proton, na? So electron remain here, proton went there. Got the idea? Understanding the idea? So it becomes COO minus and that become NH3 plus. So both positive and negative charge is existing or not here? Yes, na? So can we say this as a which form? Deuterionic form. So amino acid exists in a deuterionic form but at a specific pH, and that pH is variable from different amino acid. Now I am just moving ahead. What happened when we change the pH? If we suppose that I am changing the pH in acidic medium, what happened? in presence of H plus ion. So acidic pH means what? Lower pH than the specific pH. Don't think like it is 7 and acidic means less than 7. Suppose that here it is 5 pH, deuterionic nature sol. So less than 5 is the acidic. And more than 5 will consider here as what? Basic. Getting the idea or not? So at acidic pH, what you will find, have a look. What happened here? Ki C NH3 plus COOH H, and here which group? R group. What happened? These H plus accepted by COO minus and converted into electron was there, proton went converted into hydrogen, COOH. So it becomes which charge? Positive charge. It becomes which charge? Positive charge. So when acidic pH, getting the idea or not? Is it clear in the mind? So when you are finding about what is the specific pH, below than the specific pH, become acidic, na? Above than the specific pH, become basic from any referential. So suppose that referential is 5 pH, below than 5, acidic pH, above than 5, basic pH, we will consider like this. Getting the idea? So here it is getting which charge? Positive charge. It is getting which charge? Positive charge. Now at basic pH what happened? At basic pH what happened? OH minus is here. So what happened? Look here. CNH2 R C OO minus and H. What you find here? Here OH minus and here NH3 plus. So hydrogen is going to attach with this OH, react with this OH, and it is going to release as what? Water, and NH3 plus converted into NH2 plus. Overall charge is how much? COO minus. So which charge? Negative charge. So at basic pH, what happening? Positive charge developing. And at acidic pH, which one? Neutral charge, at acidic pH, who is developing? Positive charge. At basic pH, who is developing? Negative charge. And at a particular pH, which is there? Both positive and negative charge. But when both positive and negative charge is there, this condition is called which one? Deuterionic form of the amino acid. So in exam, in medical entrance, simply what the question they will ask? Which of the following is deuterionic form? So you just look who have both positive and negative charge. Net charge 0 is deuterionic form. Got the idea? And these deuterionic forms is formed by a particular amino acid at a specific pH. Got the idea? So this is the deuterionic form of the amino acid, and amino acid syllabus is over for this weekend. Okay, ji, thank you very much. And if any doubt, you can ask. Okay? Any query, any question? You got the deuterionic form? Both positive, negative charge when present in amino acid deuterionic form. Simple. In biology, that much only. Right? And it going to, for different amino acid, the pH for showing their deuterionic nature is different. That things keep in the mind, in the biology section only. Done? Rest in biochemistry, chemistry sir will talk about it. Is it all right? Okay, ji, bye-bye. So happy Dasara to all of you. Good night, good night, good night. I'm discontinuing the meeting. Bye.